Chancellor Hall, President Woodin, Dean Robinson, honored guests, and most importantly, you, the amazing graduates of the Temeti Faculty of Medicine, along with your family and friends who have supported you. Many congratulations on all your achievements. I'm truly honored to receive this honorary degree from the University of Toronto, where I've spent the last 40 years or so of my career. It's caused me to look back over my time in Canada and the changes I've seen in science and the application to human health. I trained in the UK at Oxford and Cambridge, and I arrived in Canada on faculty at Brock University in 1977. Just before I left Oxford, I heard a lecture from Fred Sanger about how they had sequenced the DNA genome of a bacterial virus, all 5,000 nucleotides. It was a huge achievement. There were standing ovations, people, congratulating. He received his second Nobel Prize for this technology. 1977. Fast forward to 2001, when the first draft human genome sequence, all 6 billion base pairs, was announced at the cost of maybe $2 billion. To today, when genome sequencing is so cheap and rapid that your personal genome sequence can be used to help define your future health trajectory. Amazing progress that all of you who are actually taking up careers in medicine will see to come to fruition in terms of true precision health. In my own research, I've seen major advances over the past 50 years. I'm fundamentally a basic scientist. I'm interested in how a single cell, the fertilized egg, becomes a human being. It's a simple question, not so simple answers. But along the way, we've learned so much that is relevant to human development and disease. Most importantly, we help define the origin of pluripotent stem cells, which are now being developed as a source of cells to treat many forms of human degenerative diseases. It's a very exciting time now to be a stem cell researcher. We're seeing new pluripotent stem cell-derived treatments moving rapidly forward to the clinic for diseases like macular degeneration, Parkinson's, diabetes, and heart disease. I also became a mouse geneticist along the way to understand how genes control embryo and stem cell development. With my colleagues here in Toronto at the Lunenfeld Research Institute in the 80s and 90s, we were among the first to be able to make targeted genetic alterations in a mouse to study gene function. I co-led international projects to mutate every gene in the mouse genome. Fast forward again to the 2000s and the discovery of the CRISPR-Cas9 system in yogurt bacteria that has provided an incredibly precise and efficient tool to edit genes in any cell, including mouse embryos. My project, those cells are sitting in freezers. Everybody uses CRISPR. Most excitingly, of course, we're now seeing the application of human gene editing to help treat and cure people suffering from serious genetic diseases. This all sounds wonderful, but along the way in these research domains, there are ethical and societal implications that must not be ignored. As a scientist working mostly in mouse stem cells, embryos, and gene editing, I considered it my duty to engage thoughtfully in ongoing debates about the acceptability of extension into human applications. For example, the power of gene editing has raised many concerns about the potential of moving from curing patients with diseases to editing embryos to make so-called designer babies. Over the past 10 years, as you heard, I've had the great privilege to be the president of the Gairdner Foundation, which gives out the Canada Gairdner International Awards for transformative research that impacts human health. This is one of the best jobs in science, celebrating excellence by recognizing the very best biomedical researchers in the world and bringing them here to Canada to tell their stories and inspire the next generation of scientists and innovators who will be ready to tackle the major issues facing the world today. Our laureates have included Dowden and Charpentier for CRISPR gene editing, Stu Orkin for defining how to genetically reactivate globin genes to treat sickle cell anemia, Carrico, Weissman, and Cullis for developing messenger RNAs against COVID, Welsh and Negulescu for producing novel drugs that have transformed the lives of patients with cystic fibrosis. We have an amazing gala every year where the laureates get to tell us a bit about their journey and their impact. Wonderful stories. I encourage you to check it all out on our YouTube channel. Last year, the first person in Canada to receive the new cystic fibrosis drugs was in the audience, alive and living a normal life after being close to the end of her life journey with cystic fibrosis. Incredibly emotional moment that spoke to the power of research and the dedication of researchers. But all of these advances too bring their own concerns, particularly with regard to the affordability and global accessibility of these new treatments. How can we make gene editing for sickle cell anemia available in the countries with the highest burden of disease in Africa? How can we ensure that the new designer drugs, like those for cystic fibrosis that are coming to the clinic, can be affordable in public healthcare systems like ours? By celebrating the breakthroughs, Gärtner provides a platform to raise these important issues going forward. And I encourage all of you in your future careers to always be aware of the ethical and societal context of what you do and to work towards a truly equitable healthcare system for all. At the Gärtner Foundation, we don't only focus on research in the prosperous countries of the world. The John Dirks Canada Global Health Award focuses on research with impact in the global south and provides many lessons for us all. There is so much to learn when we stop thinking that we in the global north know all the answers. Two examples from recent Gärtner Global Health awardees illustrate the point. Cesar Vitora, an epidemiologist working with indigenous populations in the very south of Brazil, developed a newborn birth cohort and demonstrated the importance of early breastfeeding for reaching developmental milestones. That study is now the basis of the WHO recommendation for babies worldwide. Vikram Patel in India developed effective community interventions in mental health where no specialty services were available. Those interventions are now being implemented in underserved populations here in North America. So where does the future of science lie? That's a tough question to answer. But what I do know is that science and evidence-based policy will be the key to addressing the global problems that face the world: climate change, future pandemics, food insecurity, mental health, social inequities, and more. I can also predict that science will continue to surprise us with new discoveries and unexpected applications, provided that we let creative scientists pursue their passion. I encourage all of you to pursue your passion, your creativity, and your inspiration wherever your career takes you. You are the ones who will be guiding the future of medicine. But not just in medicine. The global problems that face the world will require global solutions with diverse voices and skill sets at the table. You and your peers will be part of those solutions. There are many challenges ahead, but the rewards for humanity are huge. Be ready to take up the challenge and show that Canada leads. Thank you.